Dear colleagues, I'm very glad to see you all in our auditorium, and I'm happy to present our today speaker, Alexei Oskolsky, with a very rare topic for our workshop on fossil woods. I'm sure it's interesting, difficult, and it will broaden our landscapes. Please, Alexei.
Natalia, thank you very much for invitation to present our results. It's I'm very glad to attend your workshop. So, and I will tell you about the late Pleistocene woods from South China, from Maoming Basin, in the context of history of species distribution ranges of related to glacial and interglacial fluctuations in low latitudes. So, but first of all, good morning or good afternoon or good evening to everyone. And so that's these results are these results of my long collaboration with Professor Jianhua and Doctor or already Professor Luliang Huang from the University of from Sun Yat-sen University in Guangzhou. We collaborate quite a long time. And so today I will tell about the Pleistocene wood, and that's why I will let's start with a couple of words about Pleistocene. It was the epoch of rapid and intensive climatic changes. Just a moment. Sorry, do you see me?
Yeah.
I don't. Okay. Sorry. Okay.
It seems that everything is
So
okay.
that's
We see you,
okay.
we hear you.
Yes. So the Pleistocene, it was the epoch of climatic changes, of glaciation and interglacial cycles. And these oscillation climatic provide the decrease and loss of extinction of quite many thermophilic species, especially in northern hemisphere. But for some species, for cold tolerant species, the glaciation provide the more favorable conditions, and their distribution ranges of the species expanded. As a very famous example are the species with Arctic alpine range disjunction. So in Europe and in North America, there are quite many species distributed in tundra and occurring also in mountains in about several thousand kilometers southward from tundra. For instance, in Alps, European Alps on Carpathy. So this disjunction, several thousand kilometers, as a result of their history. During the last glaciation, these species were distributed in tundra or tundra steppe, which was spread from mountains to the continental ice sheet. And then they were spread had a much wider distribution than now. Then, during the Holocene warming, the ice sheet melted and the tundra moved to the north. And as a result, these species move also to the north, but some populations persist in refuge in mountains, which are in temperate zone now. For instance, in Alps. So this is this scenario of history of distribution range, for instance, for Drias, for Frazas, a beautiful flower, tundra flower, and some other tundra plants like Betulonana, Salix arctica, Ranunculus glacialis, Arabis, Veronica, and so on. And what's important that this scenario is well documented by pollen, by macrofossils, and also by molecular phylogeographics. But all this data are about the temperate zone. But what about the glacial expansions or contractions of the species ranged in tropical and subtropical zones? These data are much more poor. And as I will tell you about the Pleistocene of China, I will focus on last glacial maximum in tropical and subtropical East Asia. A couple of words, what happened in this time in this region. First, there was no ice, continental ice sheet. Another important thing that the sea level was about 110 meters lower than now. That's why the continents were wider, and the short line was different from the present, and some islands like Hainan or Taiwan were the part of continents. And the climate was somewhat cooler, and it's 2 to 4 degrees cooler than present, and in some parts up to 8 degrees colder. And the climate generally was drier, especially in eastern part of East Asia, but it was wetter in western East Asia. About the vegetation, this is the data of Lee. It's summary of pollen data for the vegetation during the last glacial maximum. And you see that there were the extensive dry grassland, the steppe, in the northern China, and desert in northwestern part. But in southern part, there were the forest vegetation, temperate deciduous forms to warm mixed forest in southern China. But important for us, there are no pollen records about the tropical forests in this region. It's important for our text. What about the glacial range expansion? It was colder. That's why for some cold-tolerant species, these events are documented. For Pinus quantunensis, for Pinus armandi, for Emenopteris henry, for Rosa sericea, but all these changes of range are documented by indirect evidence only by phylogeographic data, molecular phylogeographic data, or by modeling of the distribution ranges based on climatic reconstruction. The same we can say about the opposite scenario, glacial contraction with interglacial expansions are also documented for some species from this region, for instance, for Pinus tabuliformis, but also there are no direct paleontological confirmation of this scenario. In a present talk, I will share some results of our studies of mummified woods from the late Pleistocene of Maoming Basin. It is in Guangdong province in South China, focusing on the importance as evidence of glacial or interglacial fluctuation of species ranges. In this talk, I will tell you about our methodology. Then I present the data on 2 species. Sorry. Pinus armandi and Magnolia insignis, showing the glacial expansion of their range before and during the last glacial maximum. And then I present the data on Cryptocarya kinetis, the case of opposite scenario, range contraction in the last glaciation and post-glacial expansion of range. And finally, I would like to present my hypothesis that I formulated during the work on this presentation, that Maoming Basin can be considered as a glacial refuge of tropical vegetation during the late Pleistocene. Okay, and first, material and methods, locality. This Maoming Basin, it's located in Guangdong province. It's about 300 kilometers from Guangzhou. And in this locality, the lacustrine deposits with many well perfectly preserved mummified woods and also mummified fruits. And the age is estimated as late Pleistocene, about 33 to 30,000 years ago. So this is the chart showing the change of global temperature in the late Pleistocene and Holocene based on oxygen 18 isotope. And we can see that both the paleoflora of the condition of paleoflora of the Maoming Basin and was nearly the same as during the last glacial maximum. It was before the Holocene warming, and the climatic conditions were nearly the same as in last glacial maximum there. Okay, in methodology, actually we have to provide two kinds of evidence. First, we have to provide the evidence for change of species distribution, a different species or the same species distribution in the late Pleistocene and now. As we work with mummified wood, we have to identify this wood and we have to attribute these fossil woods to modern species. It's a challenge because usually the woods can be attributed only to genera. And only for some woods, if we are lucky, we can determine the species with high confidence. Another kinds of evidence, we have to show that if we found the changes of species distribution range, we have to show that they correlations, at least correlations, with the climatic changes, the influence of climatic factors on the distribution of the species. For that, we use the paleoclimatic reconstruction for late Pleistocene, the Maoming Basin, including our reconstruction based on other all available fossils from Maoming Basin, we estimated the ranges of 19 bioclimatic variables from world climate using the coexistence approach. And from the other side, we used the data from the dataset of oscillator and from for the world climate dataset for last glacial maximum and also for early Holocene. And also we, mostly my colleagues, use the species distributed modeling, the biomet ensemble approach. And this method is important to suggest for us. It's interesting to suggest the climatic factors of greatest influence on the changes of species range, on the contraction or expansion. And it also allows to make the map of climatically suitable habitats for the species, not only now, but also for the some points time in the late Pleistocene and Holocene. And we use our fossil data to test these results because these results are indirect. And so you see, this is the list of taxa which are known from the Maoming Basin. Most of them are woods. Some taxa, some data are published, some not. And there are also the fruits, very interesting fruits. And we were able to identify the species only for four fossil woods. Pinus armandi, Liquidambar formazana, Magnolia insignis, and Cryptocarya chinensis. For three of them, the data for three of them we will analyze and present here. So, and about the climate. of paleoclimates for Maoming Basin generally. What we can see this data from different proxies, and that 30,000 years ago and 20,000 years ago in late Pleistocene, the temperature was nearly the same in the Maoming Basin, and the temperature increased in Holocene. And about the sorry, so about the precipitation, the precipitation was lower in late Pleistocene than in Holocene, and it was a bit higher 30,000 years ago than 20,000 years ago in the last glacial maximum. And then the precipitation sharply increased in Holocene. And the opposite trend shows the precipitation seasonality. It's mostly because the precipitation of driest month was decreased during the last glacial maximum. Okay, and now I will tell you about the two species, Pinus armandi and Magnolia insignis, and their wood as evidence of glacial expansion. So, Pinus armandi is Chinese white pine, distributed in mountains of central and southwest China, and also in Taiwan. And these are the cold-tolerant mountain species. And they are the molecular phylogenetic evidence for the Pleistocene range expansion of this glacial expansion of this species, but there was no fossil evidence. Another species is Magnolia insignis. Roughly speaking, it has the same ecology and distribution. It is also from the species from mountains of southwestern China. It's from elevation occurs with elevation of 1,700 species. Also cold tolerant mountain species. Also, like the Pinus armandi, it doesn't occur now in the near the Maoming Basin. So, identification of mummified woods, I will show you this process on example of Pinus armandi wood. You see, this is the generally mummified woods are perfectly very easy to study by usual anatomical methods. We can cut it with microtome and study under light microscope. And you see, this is the micrographs of sections, anatomical sections of this wood, and this is typical conifer with the vertical and radial resin ducts having thin-walled epithelial cells. This condition, thin-walled epithelial cells, is characteristic for pines, for pinus. Then, another diagnostic character, smooth walls of ray tracheids. This is typical for the genus strobus, for white or for soft pines. Then, we found window-like cross-filled pits arranged only in one row. These window-like cross-filled pits reported only in 13 species of this subgenus. Then, in some of them, there are the cross-filled pits in two horizontal rows. In our wood, we did not find this condition. We can exclude this species. Then, in some crossfields, we found the pits, the two pits, mostly one, but in some, but not more. In some species, in 5 species, there are up to 4 pits on crossfields. We exclude this species. Then, very good character, smooth end walls of ray parenchyma cells, not tracheids, but parenchyma cells. In many species, there are the nodular end walls of ray parenchyma cells. we can exclude them. Then, in our wood, we found the fusiform rays. This is the rays containing the resin ducts, which are up to narrow. They are up to 2 cells in width. They are biseriate. In most species, these rays are more than 2 biseriate, up to 4 cells in width. We can exclude, and also 2 other species, Pinus pumila and Pinus siberica, they characterized by exclusively uniseriate pitching on radial tracheids walls. In our case, we can see on this picture the pits on radial walls can be in biseriate order arrangement. So, and after this, comparing all this data, we suggest that this species is Pinus armandi. And the same methodology we used for identification of Magnolia insignis. I will not tell you about it. This is published results. I can. What is important to say that for us it's important that both genera, Magnolia and Pinus, are well explored by wood anatomists. And attribution of fossil wood to modern species would be impossible if these genera were not were underexplored. So now results of species distribution modeling of the range of Pinus armadi. We mostly use the maximum maxent algorithm. And on this, actually, you have to understand that this is not maps of this species distribution. These maps show the areas where with climatically suitable habitats, the areas where there are no climatic obstacles for presence of the species. It doesn't mean that species are necessarily present in this area. Nevertheless, this method is quite useful. And we can see the red arrow shows the local fossil locality. And in the late Pleistocene, in this locality, we had it was suitable to more or less suitable, not highly suitable, but more or less suitable for this Pinus armadi. And then in Holocene, we can see that the range of these suitable habitats was contracted. And this modeling suggests that the most important characters influencing on the range of the species, Pinus armandi, are minimum winter temperature and maximum summer temperature. It is perfectly consistent with the known data on evolution or changes of climate from Holocene or from late Pleistocene to Holocene. And roughly speaking, it was too warm winter and too hot summers in Holocene for this species. And the same story with Magnolia, and we can see even that in Maoming we had a highly suitable habitat for this species in late Pleistocene, and then this area moved from this region, and the difference is that for this species, it is important not only maximum summer temperature, but also the precipitation of driest month and driest quarter. So now it is too much precipitation, and and we can estimate why these two species are not distributed now don't occur in Maoming area. And we estimated the ranges of 19th bioclimatic variables for Pinus armandi and compare it with the um, values of these bioclimatic variables for the late Pleistocene. It is white ring, and for modern time it is yellow squares. And we can see that yellow squares have critical values for the species for mean diurnal range and for which is too large now, and for minimum temperature minimum temperature of coldest month, mean temperature of wettest quarter, and mean temperature of warmest quarter. So this the temperature are too high now for this species. The same characters are typical for the we found for the same results we found for Magnolia insignis. So actually, the distribution of this species in the lack of the species in modern Maoming Basin depends on the temperature, not of the not of precipitation. Therefore, we can see that mummified woods of these two species found in Maoming Basin indicate that these species had wider distributions 30,000 years ago than now. And our climatic analysis suggests that distributions of these species were contracted due to the general warming during the Holocene. Thus, we can consider these mummified woods as the first known macrofossil evidence for glacial expansions of cold-tolerant species in low latitudes. Then, next story about Cryptocarya, showing as a case of a opposite range, opposite scenario, range contraction during the glaciation and post-glacial glacial expansion. So Cryptocarya, unlike the previous species, Cryptocarya is thermophilic species. This is the tree from subtropical forest of South China and Taiwan, and it can be found in Maoming Basin now. And a couple of words about Cryptocarya, about the genus. This is the largest genus, one of the largest genera of Lauraceae family. It's distributed worldwide in tropical and subtropical region, excluding equatorial Africa. But the center of its modern diversity in Southeast Asia. But molecular data suggests the Gondwanian origin of this genus. But the fossil record is very poor. When we studied the Cryptocarya hinensis, it was the first known fossil of this genus in Asia. But recently, from my colleagues in Sichuan Botanical Garden and in Chengdu, we described the wood, another Cryptocarya wood, from the Miocene of Yunnan, Cryptocarya lateradiata. And also, independently of us, the flower of Cryptocarya found in the Miocene of eastern China in Fujian. So this data suggests the Cryptocarya, probably the Cryptocarya can penetrate into the into the China in Eastern Asia from Australia between early Miocene or late Oligocene, when this was the time when the Indochina and island in between arose, and the bridges are formed. But it is just the hypothesis. Okay, and about the Cryptocarya hinensis. So, how did we identify it? So, first, this wood showed the combination of traits typical for Lauraceae. Simple perforation plates, alternate intervessel pits, oil or mucilage cells associated with rays, and also important character of the vessel ray pits with quite large pits with reduced borders. If we add some characters of parenchyma, axial parenchyma, lack of confluent parenchyma, but presence of marginal parenchyma, and lack of crystals, we can this combination of characters found only in Cryptocarya. But to determine the affinity of this fossil wood to extant species, we need to compare it with fossil, but the published data are quite poor. Only 3 species have been of Asian Cryptocarya have been studied before. I mean the wood anatomy of the species. And we examined the wood structure of 15 modern Cryptocarya species from China and Malaysia, of 15 of 21, as far as I remember. So, and then having this data set, we compared our wood with woods of other Cryptocarya species, and this combination, some additional characters like size of intervessel pits, and quite specific feature, the presence of aggregate rays is quite uncommon feature, suggests that this species, this wood belongs to Cryptocarya chinensis. Okay, what's interesting that this wood contain the pit flags, and these pit flags have a quite characteristic shape, and which typical for mines, cambium mines, result of activity of cambium mining larvae of fly of Diptera, fly of Phytobia genus. And the mines of Phytobia found in 13 genera of Lauratia, but have never been reported in Cryptocarya. We found it in Cryptocarya. Okay, then about the Cryptocarya is interesting for us because this is Thermophilus genus, and Cryptocarya species not found in region with frosty climate. For Cryptocarya chinensis, one of the most northern species of this genus, the range between of cold minimum temperature of coldest month varies from 1.2 degrees to 17.1 degrees. And it means it's the evidence of frostless climate in the Maoming Basin during the last glaciation, at least for this time, 30 to 33,000 years ago. Then, this data of Cryptocarya, the results of Maxent modeling of suitable habitats for Cryptocarya chinensis in different time. So, and we can see that the region of Maoming was suitable for Cryptocarya chinensis in the late Pleistocene, including 21,000 years ago at the last glacial maximum. So, what is and these result of this analysis are confirmed by our fossil evidence. But you can see that the region, these areas of suitable habitats were very restricted, especially in the late glacial maximum. It was several spots along the but it was not widely very widely distributed area. Okay, widely expanded area. And in modern condition, the suitable habitats of the species are widely distributed, you can see. And so then we can. It's highly likely that the range of cryptocaryokinensis underwent the contraction during the late Pleistocene and expansion in the Holocene. And if this scenario is true, it's highly likely that Maoming Basin is a relic locality of cryptocaryokinensis, and the species probably persist there at least since the late Pleistocene. And these are the bioclimatic parameters of greatest influence on distribution of this species. And we can see that the most important are the variation of temporal variation of temperature. It's diurnal range and temporal seasonality and isothermality. And also the precipitation of warmest and coldest quarter are also does matter. So generally, this data suggested that the range of cryptocaryokinensis in the late Pleistocene was restricted due to continental aridity and higher temperature seasonality in this region. And in Holocene, we can see the shift of more humid climate and decrease in temperature seasonality in Holocene. And these changes in climate probably associated with weakening of winter monsoon and intensification of summer monsoon. Its changes are typical for the shift from glacial to interglacial regime. And these changes are probably influence or have influence on range expansion of cryptocaryokinensis. So, and these actually the maps of reconstructed maps of distribution of suitable habitats of cryptocarya suggested me for some to consider the role of Maoming Basin in the history of vegetation of this region. You see, I repeat this map based on pollen data. This is the reconstruction of main biomes in vegetation of China during the last glacial maximum. We can see that in region of Maoming Basin, there were covered by broad leaves, evergreen to warm mixed forest, according to the pollen data, pollen record. But there are no pollen records for the tropical forests in China during this time. But in our flora, the Maoming Basin flora, we can find several species, quite many species, which are typical for warm mixed forests, suggesting this type of vegetation, this biome. It is Pinus armandi, Magnolia insignis, Cataleria, and so on. But what is more important that we found several tropical taxa. For instance, Canarium, probably Sapotacea wood, probably Erioflare. Probably we can interpret Cryptocarya also as tropical forest plant, but I'm not sure its interpretation. But in any case, it is thermophilous species. So our results partly support the data of palynological data, palynological records of this wood work of Lee with co-author. But also we can see the presence of plants from tropical forest in the Maoming Basin. And although the Maoming flora existed before the last glacial maximum, its reconstruction of this climate, the values of its climatic parameters are nearly the same as in the same place during the last glacial maximum. My question is, if the tropical taxa would be able to survive in that climate, in the late Pleistocene of Maoming Basin. And to answer this question, I made such an experiment. I checked the climatic ranges of Canarium pimela. Why Canarium? Because the fruits of Canarium have been described from Maoming Basin by Huang and co-authors 3 years ago. And these fruits, unfortunately, they were not attributed to any modern species, but no doubt that this is Canarium. But Canarium represents a tropical evergreen functional plant type. This is definitely a tropical plant. And I took the Canarium pimela as I selected it because this species are now found in the vicinity of Maoming Basin. And this is the ranges of bioclimatic variables within the distribution area of Canarium pimela, and I compare it with the reconstructed data of these variables for 30,000 years ago and for 20,000 years ago. First are green squares, another are red squares. Red squares are last glacial matching. And you see all these values are within the ranges of in which this species exist now, occurs now. So we can say that Canarium pinella was able to survive during the last glaciation in Maoming Basin. If so, we can hypothesize that Maoming Basin was a glacial refuge for tropical vegetation during the late Pleistocene. Of course, this is just the hypothesis, but we can see some evidence, like the presence of tropical macrofossils in the Maoming Basin in the late Pleistocene. Second, this paleoclimatic reconstruction suggests that at least some tropical plants can live would be able to live in these conditions. And please also look on the map reconstruction of climatically suitable habitats for Cryptocarya chinensis, the thermophilic species. It was very restricted. Probably it was very restricted refuge, refuge for this vegetation, and that's why the tropical taxa lack in the pollen records from last glacial maximum from this region. And of course, I don't know why this refuge exists, which climatic or geological factors influence on this. It is also interesting question, but this is just the hypothesis, and we need to study other woods and roots from Maoming Basin and to analyze the ecological ranges and to make the models of the distribution to test this hypothesis. Okay, so to conclude, we can have the mummified woods of two modern species from Maoming Basin, Pinus armadi and Magnolia insignis, suggesting that 30 years ago they have wider distribution than now. And distribution of the species contracted due to warming during the Holocene. That's why these woods can be considered as the macrofossil evidence of glacial expansion and interglacial glacial expansion of cold-tolerant species in low latitudes. Then mummified wood of Cryptocarya chinensis suggest the presence in this flora suggests that this species persists in Maoming Basin since late Pleistocene. Maoming Basin is a relic area for this species, and it's highly likely that the species underwent post-glacial expansion during the Holocene. And the late Pleistocene flora of Maoming Basin represent mostly the plants of warm mixed forest, but some taxa contain some taxa of tropical forest biomes. And the data suggest that climate of Maoming Basin during the last glaciation was suitable, at least for some tropical taxa. That's why we can hypothesize that Maoming Basin was the small glacial refuge of tropical vegetation during the last glaciation. Okay, so no, results of our study have been published in these three papers. So, and we are I'm glad to say that two of these papers have been included into top 10 of achievement of Chinese paleontology in 2023. And it's funny that the subject of our collaboration have been translated by Google as "speak lowly of the season when her husband Jose comes to love her." Okay, thank you very much for your attention. Сессия, спасибо за внимание.
Thank you, Alexei, for such a multifaceted talk. Really, thank you. It was impressive. And now,
Thank you, thank you.
time for questions. If anybody wants to ask question, let's show your hand like this, or show the artificial hand, or write your question in the chat, and I will read it for everybody as you prefer.
Okay, I need to open the chat. Okay, thank you.
Well, meantime, I'd like to pose my small question, but I'm curious too. How did these insects enter the wood? How they manage?
Well, I think very simple through the bark, and they so honestly saying, I don't know the mechanism. But this insect, this flag, is arranged along the boundary of growth ring. It means that it was infected in early in the spring, in the very beginning of vegetation season, or who knows, maybe it was infected in previous year, but the egg was put in this cambium. So, but anyway, it is maybe the entomologist can answer. I don't know.
Well, I see a question from just a moment. You may also look in the chat.
Yes.
I see two questions. The first one from Shanbir Davids. Can these taxa serve as ecological indicators of paleoclimate phases for Asia?
I would say that it is early to say that these taxa serve as ecological indicators, but there is for us it's important that these taxa exist now, and now we can analyze their ecological niche. We have the data for the ecological niche, and then on this basis, we can reconstruct ecological niche and ecological effect in the past. It's just quite a rare opportunity when we have the modern species, not genera, not family, the species, the same species in fossil condition. That's why we can use the methods of modern ecology for to analyze the climate and distribution in the past.
Thank you. It's really fascinating to deal with modern species in fossil state. Well, the second question from the chat from Arun Joshi. Congratulations, Sir, for such a nice presentation. I would like to know, how do fusiform initial cells of fossil woods are different from living woody plants, and how they can be helpful to understand species distribution or vegetation type in the past and present scenarios?
Okay, first, fusiform
initials of cells.
Yes. So I answered the first question. And actually, we didn't observe the fusiform initials. We observed the wood. And in wood, the derivatives of the cells derived from fusiform initials have lignified secondary walls. That's why the cells with secondary walls are quite well preserved in fossils, especially in mummified fossils. Another question, it's about uh, do you mean how the fusiform the cells can help to understand the species distribution? So if I correctly understand your question, the characteristics of a cell of wood help us to identify them. And if we correctly identify, convincingly identify this wood, then we can reconstruct the distributions and so on the basis of our knowledge of and our data on modern distribution of these species.
Mhm. Thank you. The next question is from Anna Stepanova. How many samples of mummified Pinus armandi were in the study?
Mummified, as far as I know, it was one large sample.
Mhm. And the next question is not question, but a comment, but you perhaps will like to comment it as well. Also, it is the same species. We are not sure that living species have the same genes or allele than fossil specimens.
Okay. Okay, it is correct, but the same objection can be is valid for any ecological study. If we study the same species in the another in another population, we are not sure that they have the same genes. That's why, of course, it's possible, but it is how to say it's presumption of ecological studies that they are more or less the same in genetics, in structure, and in its adaptive characters and adaptive strategy. So, thank you
Binar,
very
have you
okay.
satisfied with the answer? Yeah. Well, and Arun Joshi also writes you, thank you very much for answering the question.
Thank you.
Well, do we have more questions to the speaker? I don't see hands. Well, I'd like to pose one more question if you don't mind.
Yeah, please.
You told us about modeling that reconstructs suitable areas where a given plant could theoretically grow. How much is the difference between these theoretical areas and the real areas where the plant grows?
Usually, these theoretical areas are wider than real. It's like the difference between predicted and real ecological niche, because the distribution of species depends not only of climate, but this is the prediction based on climate. As far as I know, there are some methods which also can estimate it on can use not only climatic data but also the edaphic soil data. But we did not use it. We don't have any information about soil. So of course it is wider, and we can see that even for Cryptocarya chinensis that the predicted region for now is wider than real distribution area, for instance.
Thank you.
Of course, this is indirect predictions. It's based on indirect data, but for me it's more interesting to formulate the question for further. We hope it helps to formulate the question for to search for direct paleontological confirmation or refutation of these ideas.
Dig there.
Yeah.
Go and search. And I see hand Tatiana Faraponova. You may switch on your microphone and ask.
Thank you. Thank you for your talk. I'm curious if we find modern species in fossils, we know their ecological preferences, and if these modern species are found together with fossil species, as I understand, we can predict their ecological preferences to some degree. And how accurate are these predictions?
It's a good question, but there it is generally there is the presumption of ecological niche conservatism. That's the evolutionary ecological niches, including climatic niches, are quite conservative in their evolution. So, and I'm personally, I'm quite skeptical when these ideas are used for reconstruction of ecological reconstruction, for instance, for Miocene and all that. But now we have quite the species, quite young species. It's only 30,000 years. It is not the time for essential changes in the evolution. I think this is the case when we can use it. There is no essential. I don't think that the species undergone essential and prevent essential changes in their ecology. Of course, there is the place for skepticism, but okay.
Okay, thank you.
Thank you, Alexei. Do we have more questions to the speaker? Last chance to pose your question. Okay. A comment from Gunnar. Some species can change of allele quite quickly.
Okay. And for us, it's interesting. Not genetics but ecology. Are they able to change the ecological, the adaptive strategy, the ecological strategies, and so on, which are more important for in the context of this study than their genetics. Of course, the mutation occurs permanently, and no problem. But it is quite difficult to discuss this matter in the terms of genes.
And addition to this comment, it is at least true for some experimental angiosperms. Well, this is again about the...
Yes, sure. It would be great to find the fossil of Arabidopsis somewhere in Pleistocene or Miocene. It would be great.
And one comment, nice talk and nice to meet you all on net.
Thank you. Me too.
One more last call for the questions. It seems that no more questions. Thank you, Alexei, for a very interesting talk. Thank you all for coming.
And thank you very much for invitation. Thank you for a very interesting discussion. So that's I hope maybe if you are interested in some collaboration, you are welcome to contact with me. Okay, thank you.